Welcome back, everyone. I hope you had a chance to stretch and refresh. We are now moving into the Session 2 Q&A panel discussion, where we will dive deeper into the topics covered in the presentations and address your questions. As a reminder, you may continue to submit your questions through the MS Teams chat Q&A function or via email to orsevents@fda.hhs.gov with the subject line of AGDD 2025, and we will address as many questions as possible. Please welcome back Dr. Zhang, Dr. Wu, Dr. Chung, Dr. Nguyen, and Dr. Gong to our panel. In addition to our previous speakers, we would like to welcome 2 additional panelists. We have Dr. Vivian, who serves as an Associate Division Director in the Division of Bioequivalence 2 in the Office of Bioequivalence, Office of Generic Drugs. And welcome Dr. Wanjie Sun, who serves as a Master Mathematical Statistician in the Division of Biometrics 8, Office of Biostatistics, Office of Translational Sciences. Thank you to all of our panelists, and we will now take our first question.
Thank you. So we have our first question for Dr. Min. For the drugs with a long elimination half-life, T50 greater than 24 hours, can we use modeling to predict residual concentrations and subtract these predicted residual concentrations from the subsequent period's observation instead of implementing sufficient washout?
Thank you for the question. For this question, typically applicants are required to implement adequate washout period of at least 5 elimination half-life. And for modeling, it can be used to support the scenarios that inadequate washout period, when extended washout period is not feasible due to some study logistics, or like the case we discussed in our presentation, carryover still occurs despite having an adequate washout period. For this model-based approach, we want to emphasize that a comprehensive model validation and evaluation is required. Thank you.
Thank you, Dr. Min. So we have our next question for Dr. Vivian and Dr. Fang Wu. What data could be used to support or justify the BCS3-based biowaiver for generic drug with API that has more than 10% degradation at acidic pH?
Thanks. So I think Dr. Fang and Dr. Usha gave a good case example of one case where we considered this for BCS-based biowaiver, even though it's slightly different than what's in the ICH M9 guidance. One thing that I think is important is that the BCS guidance, the in vitro studies exist to really surrogate an in vivo BE to make sure that there's low risk for in vivo bioinequivalence. So one important piece of data I think here was that we had some in vivo information from literature and other sources to say, you know, this may be happening in vitro, but we're seeing in these PPI studies and in a FEDS study, there may not be an impact in vivo on differences in gastric pH. So here we were seeing the degradation at low pH. The in vivo data suggested maybe for this particular drug, it didn't have an impact. And of course, there was also some additional data we asked to reduce the risk even further, such as looking at the degradation comparison in different pH and different dissolution between the test and reference products to ensure it was comparable. And also supportive modeling was helpful. I think Fang can tell you a little more about that.
Yeah, so as we introduced in presentation, that we use the modeling and can identify the FASTGIF as a biopredictive media, that by incorporating the in vitro dissolution in FASTGIF medium and also the degradation data, we can predict that even with the degradation, but if it's with a similar rate and degree between the generic drug and the RLD, there will be a low risk of in vivo non-BE, even with the degradation. Thank you.
Thank you, Dr. Vivian, and thank you, Dr. Wu. We have another question for Dr. Duyen. Nguyen, the presentation shows example where patient-based PSGs were reevaluated and changed to healthy subject studies. What triggers would prompt a reassessment of existing PSGs, and how frequently does this occur?
Thank you for the question. So the reassessment of patient-based PSGs can be triggered by multiple factors, including internal and external requests. So external requests can come from either the industry or the public via communications such as control correspondences, product development meeting requests, as well as from other agencies through global harmonizations. And then internally within the FDA, we also have emerging safety data from the post-marketing space, as well as improved understanding of the mechanism of toxicity of the drug products. So based on these factors, because these requests are generally inconsistent in nature, there is no predetermined frequency for the reassessment of patient-based PSGs.
Thank you, Dr. Nguyen. We have another question for Dr. Yuqing Gong. What criteria should guide the selection of an appropriate alternative comparator? And are there situations where this extrapolation approach would not be suitable or recommended?
Thank you for the question. So as I presented, this approach will take relative ratio from the R&D and that alternative comparator based on historical data. So that historical data must be available to build such a bridge. And I also want to mention to the applicant to be mindful to consider sometimes the generic require additional study that R&D in that historical data they may not have. For example, sometimes generic recommend both fasting and fat study, but in some cases the original R&D during the NDA submission they may not have done such a study. So the applicant needs to demonstrate to us if they want to use this alternative approach, then what kind of BE criteria they are following, how they are going to design that study. Thank you.
Thank you, Dr. Gong. Our next question is for Dr. Wanjie Sun. What are the key scientific principles that allow for valid extrapolation when the RLD is not bioequivalent to the alternative comparator?
Thank you for the question. Yuqing introduced the two cases in her presentation. In case one, so basically RLD and the comparator RS, they are bioequivalent. Then the RS can be used as the comparator directly to establish bioequivalence with test product. So there are two assumptions behind this. One is the constancy assumption, as Yuqing introduced. And then the second assumption is the bioequivalence transitivity, as Yuqing discussed. Then in case two, if the RLD and the alternative comparator, if they're not bioequivalent, so in this case, if the alternative comparator is the same between the historical study and the new study, meaning that they have the same strength, same dose form, same route of administration, then we actually only need one assumption by using the bridging method we recommended for the trazodone PSG that Yuqing walked through step by step. So in this case, we actually only need the first assumption, the constancy assumption. We actually do not really need the second bioequivalence transitivity assumption. So the bridging method we introduced here actually requires fewer assumptions than the first case, so which is good news. But with this said, in practice, different applications may have different complications, different challenges. For example, the comparator in the historical study, they may use a different strength than the new study. So due to different considerations like safety, ethics, concerns. In this case, additional assumptions may be required. For example, dose proportionality. Well, all of this, they need to be approved mathematically. So the bridging statistical method we introduced in the trazodone PSG is just a general framework that can serve as a basis it may need to be adapted or tailored to fit each specific application. So sponsors, if you intend to use bridging method for bioequivalence assessment, we recommend you engaging with the agency early so we can help you tailor the bridging method to a specific application. Thank you.
Thank you, Dr. Sun. Our next question is for Commander Yi Zhang. Can literature data or alternative scientific justification for solubility be used as pivotal data to qualify a drug substance for a BCS-based biowaiver?
Thanks for the question. So the short answer is no, because you should conduct your own solubility study to establish the high solubility of the drug substances and submit it as a pivotal data. However, the literature data may be submitted as a supportive. For permeability data, you may refer to either NDA or any public available information. But back to the solubility, you have to conduct your own study across multimedia pH. Thank you.
Thank you, Commander Zhang. We have our next question for Dr. Fang Wu. Generally, what could be the pathways of communication if the applicant would like to use PBPK modeling to support BCS-based biowaiver when under certain situations that do not meet M9 basic requirements?
Thank you for the question. If the sponsors would like to use PBPK modeling to support BCS-based biowaiver beyond M9 and including cases like non-Q1, Q2, non-very rapid release APIs with degradation, you are encouraged to communicate with agency through pre-ANDA meeting communication pathways such as MIE model integrated evidence meetings or pre-ANDA meetings. Thank you.
Thank you, Dr. Wu. Our next question is for Dr. Nguyen. How do we balance the need to maintain study integrity with subject safety and retention when using antiemetics?
Thank you for this question. So I mentioned in the presentation a few recommended consideration factor when implementing antiemetics in BKB studies, including the low risk of PK interaction with the study drug via multiple channels that should be considered, but also low risk of bioanalytical interference with the study drug. And then we also recommend non-oral routes of administration for cases where the study drugs are of oral route. So when any additional considerations that the investigators can think of that can be adequately justified or welcome. In addition, we also, investigators may also consider the basis of administration. So if the antiemetics are administered prophylactically, so all subjects will receive the same antiemetic versus on an as-needed basis, that can also help further strengthen the study integrity. Thank you.
Thank you, Dr. Nguyen. Our next question is for Dr. Min Chung. You emphasized that the importance of the model accuracy. What sampling duration is sufficient to ensure the elimination phase is adequately captured and provide robust data for pharmacokinetic modeling, enabling precise and accurate characterization of the terminal elimination phase?
For PK sampling duration, our expectation is to it should be continued for at least 3 to 5 half-lives, and it should be able to capture complete absorption phase of the drug for NCA. In our presentation, we focus on immediate release drug products. For modified release drug products, the sampling duration may need to be longer in order to capture complete drug release profile. Thank you.
Can I add one comment here? So regarding modeling to address those inadequate washout issue, OGD has a GDUFA funded research our collaborator, Uppsala University, Andy Hooker, and Matt Carson's group, they demonstrated the sampling duration may be shorter if the applicant can pre-specify they are going to leverage modeling. So I think if you predefine a scientifically sound method, this duration can be shorter than the standard criteria like Dr. Ming just mentioned. So there is a poster published by them where they demonstrated the sampling duration can be much shorter, like 3 times shorter compared to the standard one. But you need to predefine if you plan to use such a modeling approach to demonstrate BE. Thank you.
Thank you, Dr. Chung, and thank you, Dr. Gong. Our next question is for Commander Yi Zhang. Can a BCS-based biowaiver for one product strength be extended to the other strengths in the product line?
Thanks for the question. No, because as I elaborated in my presentation, BCS3 based biowaiver for one strength cannot be extended to the other strengths in its product line. The BCS based biowaiver criteria, especially for 3, should be supported for each strength in its product line, including like a formulation similarity for each corresponding strengths, the in vitro dissolution comparison for each corresponding strengths of the test to its corresponding strengths in the reference product line. That matching profile will ensure there is no potential drift occur when an additional waiver is granted without a comparison to the corresponding reference product strengths. Thank you.
Thank you, Commander Zhang. Our next question is for Dr. Gong and Dr. Wanjie Sun. Given the recommendations for early FDA engagement through the Model Integrated Evidence Industry Meeting Pilot, what type of the preliminary data or analysis should sponsor prepare before initiating these discussions?
So I think we expect the applicant to give us some preliminary data regarding their generic product program, including the formulation design and any preliminary data they have, like how the study will be performed, like which data they are going to rely on to build such a scientific bridge to use alternative comparator. And ideally, they should also propose what is the equation, mathematical equation they're relying on. And I'll pass to Dr. Sun to add on that part.
Yeah, so in practice, things can be complicated. For example, the historical data, a historical study in the published literature, there may not be complete summary statistics that that would be needed based on the PSG, for trazodone, we provided. So you need the point estimate of GMR, you need mean squared error, you need the degree freedom. In published literature, you may not find those information. So you should just engage with us, and so we can help you to revise the formula, and just to and then other times there could be the RLD product and the RLD comparator, they may both are discontinued. And in this case, you may need to introduce a second alternative comparator, like the RS comparator. And then in this case, the method that we recommended in the PSG for trazodone may not be sufficient. So more complications like this may happen. So we recommend you engaging with us early. Thank you.
Thank you, Dr. Gong, and thank you, Dr. Sun. Our next question is for Dr. Diana Vivian and Dr. Fang Wu. For the cladribine case study, it was mentioned using FASSGF at pH 1.6 in addition to the standard 0.1 HCl at pH 1.2 for dissolution testing. Can you elaborate on the scientific justification for this media selection?
Sure, yes. So this media was selected for its biorelevance as a simulated gastric fluid and also for its biopredictive nature, and that was really driven by DQMM. We also thought it was helpful to have dissolution in multiple pH conditions in that kind of pH sensitive range to degradation in this case. We had multiple new issues for this drug product, one being the stability that we've mentioned, and also that the API existed as an API complex with cyclodextrin. So having that additional information with dissolution and solubility at multiple pH conditions in that sensitive range to degradation helped us to minimize risk and be sure that the test product is behaving similarly to the reference product in terms of degradation rate, dissolution, behavior of the API complex, and also the API. So all of that was just good information for us to have. And Fang can talk about more about the biopredictive nature of that.
Yeah. As Vivian mentioned, that we did use modeling and to identify what conditions are biopredictive. Actually, we incorporated the dissolution and the degradation data, dissolution in a FASTGIVE versus at pH 1.2 into the PBPK modeling. Then we found the dissolution at the FASTGIVE conditions are more biopredictive. It can predict the PK well under the fasting condition. That's why it supports that using FASTGIVE as medium for dissolution testing. Although generally 1.2, 4.5, and 6.8 are recommended for evaluation for the solubility and dissolution testing per M9 guideline. Thank you.
Thank you, Dr. Vivian, and thank you, Dr. Wu. Our next question is for Dr. Nguyen. With over 390 drugs containing PGX information in the FDA labeling, but only 17 PSGs including PGS recommendation, what are the barriers to the broader implementation? How can we prioritize which drugs should incorporate PGX considerations?
Thank you for the question. So some of the barriers for broader implementation of PGX information in PSGs include that currently the determination of the applicability is still determined on a case-by-case basis with the underlying uncertainty in the applicability to the a single-dose PKBE studies as opposed to a more well-established clinical practice. In addition, there is also a lack of dedicated studies that were specifically designed to detect differences in PK or safety pertaining to the different generic variations. A few of the prioritization criteria that may come in handy can include those that have more well-established evidence for gene-drug pairs, as well as those have more well-established evidence for PK or clinical significance. And other prioritization criteria may include those that have higher risk for more severe intensity of adverse events in healthy subjects, or cases where there are certain for example, when the study are being conducted in certain regional in the world, if the study investigator can predict higher risk of certain generic variations in those studies, those can be considered when the studies are being conducted.
Thank you, Dr. Nguyen. Our next question is for Dr. Min Chung. Based on the case scenario presented today, what other strategies that may be used to mitigate the impact of the inadequate washout in BE studies?
Thank you for the question. So based on our regulatory review experience, we do see some cases that treating period 1 data as an independent parallel study when residual concentrations are observed in the subsequent study periods. However, this approach may only apply on case-by-case basis. And we want to emphasize that our preferred approach is to implement adequate washout period to minimize carryover effect and also to ensure study validity. Thank you.
Thank you, Dr. Chung. Our next question is for Commander Zhang. For fixed dose combination products, can one of the drug substances qualified for BCS-based biowaiver while the others do not?
Thanks for the question. So for the FDC drug product, basically the similar principle applied to APIs in the FDC product. So in order to be eligible for BCS waiver, is either 1 or 3, all of the APIs in the FDC drug product have to meet the criteria per ICHM-9. You can't just say, well, my API 1 is BCS 1, but the other didn't. So, or the other additional API is 3. But as long as all of the APIs included in this fixed dose combination drug product meet the criteria delineated in the SHM9 guidance, it can be eligible for BCS. But the most important thing you have to remember, all of them have to meet, not just one or couple. Thank you.
Thank you, Commander Zhang. Our next question is for Dr. Nguyen. Should there be standardized protocols for antiemetic use across high emetogenic drugs? Or should each PSD be developed individually based on the specific drug's profile? What criteria should guide the selection of the specific antiemetic agents?
Thank you for this question. So currently, the PSGs do not include a specific recommendation for antiemetics other than that antiemetics are recommended or treatment for nausea and vomiting are recommended for those specific drug products. As stated previously, the study protocol should specify the study drugs that are selected based on the considerations that I covered in the presentation. So with that being said, the PSG do not go into the specific antiemetic or administration method being recommended to leave room for the investigators to design accordingly based on their preferred method or existing protocols at the study sites.
Thank you, Dr. Nguyen. Our next question is for Dr. Diana Vivian. If there is less than 85% of the undegraded drug substance in the dissolution study, can very rapid dissolution still be demonstrated?
Thanks. So I think the first thing to consider is, why are you seeing degradation? Do we have any concern that the stability has any impact on B in vivo? For this case, since we had already addressed the risk of the stability, we did say that it was okay as long as the degradation products were quantified and measured, and total of degradants and the parent were above 85%. So the purpose of in vitro dissolution testing is really to make sure that there is comparable release of the drug from the test and reference product. So here we were able to evaluate comparable release just by including the known and quantified degradants as well as the parent.
Thank you, Dr. Vivian. Our next question is for Dr. Min Chung. Are there scenarios where MIE-based approach cannot be applied for carryover effect adjustment? Could you please elaborate with specific examples?
So for this model-based approach is most readily applicable for immediate release drug products, which we discussed in our presentation, because immediate release drug products, their elimination is mainly determined by systemic clearance rather than formulation. For other certain formulations, such as modified release or long-acting injectables. So for those type of drug products, they may need more careful or case-by-case assessment. So like long-acting injectables, sometimes even their pre-dose concentrations are less than 5% of the respective Cmax. It can be concerning because the residual concentrations may only reflect ongoing drug release rather than the elimination process. And also, for this type of drug products, the drug release may not be complete within 5 elimination half-lives, which can create some carryover concerns. So in that scenario, using empirical PK modeling to characterize a PK profile may be more challenging. So if if applicants want have an alternative approach for this scenario, complex scenario, and for drug products with long half-life, we encourage applicants to have a discussion with us early during the drug products development. Thank you.
Thank you, Dr. Chung. Our next question is for Commander Zhang. Are drug substances that exhibit nonlinear pharmacokinetics eligible for BCS-based biowaiver?
Yes, as long as the drug substances, they meet both permeability, solubility for BCS1, and also for the solubility, very rapid dissolution, and the formulations Q1 the same, Q2 similar, those criteria, they are still eligible for BCS waiver. Thank you.
Thank you, Commander Zhang. We have another question for you again. What are the orally why are the orally disintegrating tablets not eligible for BCS-based biowaiver if they are administered without water?
Thanks for the question. So basically, if you look at the ICH-MNI guidance, it's very clearly the BCH-based biowaiver approach is based on the mode of administration of the medication with water. So that's why the ODT or buccal or sublingual tabs, they are not eligible for the BCS-based biowaiver. So I can share some thoughts from my own. Basically, we all know the residual gastric volume is well below 250 milliliter as we conducted for the solubility study. Thus, therefore, the estimation of solubility of the drug substances in 250 millimeter of liquid media is not applicable to the product that they're taking without water. So defining the volume of the media required to establish the solubility classification would be very challenging for ODT. If they are taking without water. In addition, the current dissolution methodology with very limited volume of the medium for the product that's it's a very challenge as well. Therefore, for ODT or the buccal tablet or sublingual, they are not eligible for BCS-based biowaiver. Thank you.
All right, I think we've come to the end of all of our submitted questions. So thank you to all of our Session 2 speakers and panelists for sharing your expertise and insights during today's discussion. Your contributions have provided valuable guidance on novel bioequivalent study designs and applications.